in our series. I'm just having here an issue. Just a second. Okay, so thank you everyone for being here today. A special thank you to Dr. Stefanie Schirmeier, who accepted our invitation to give a very interesting talk that will tell us about metabolism in the brain. I think everyone is going to enjoy it. So while we wait a little bit more for everyone to show up, you might have noticed that we have a poll ongoing here. So last week we were probing our community to understand from the brain-body interactions axis where they would position themselves. So today we change a little bit the focus, and we would like to know which organism do you not work on, but would actually be interested to adopt. We all know that different organisms have different strengths, and I mean, we cannot work on all of them, but yeah, so we are looking forward to hearing your opinion on this. So I would also like to let you know that we have a mailing list that you can subscribe to. So I will drop the form in the meantime, the link in the chat in the meantime, and you just have to fill in a very small form, and feel free to send it to anyone you think might be interested in getting our emails and keeping up with the talks that we will have every week. At usually this time. Okay, so let me just find that. Okay. 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 Sorry, I'm having a little bit, my internet is not collaborating a lot. Don't know what is happening. Okay, should be ready now. Okay, here it is. Oops, no, this is not it. I'm so sorry. Okay, so there you go. So in case you don't know or you are not keeping up and you just happen to pop by here today for this amazing talk, I mean, feel free to subscribe and we'll keep you updated. So I think we have some people already here. Might wait a couple more minutes. So don't forget the poll, guys. Actually, when I was trying to find which options would be interesting for people, it's quite difficult. I hope no one gets mad with us if we happen to miss the animal model or the organism that you find more exciting. Let us know in the chat in case we missed it, and which are your reasons to actually wanting to work with it. Okay. So I think we can start. So I'm going to introduce Dr. Stefanie. So Dr. Stefanie, she studied biochemistry for undergrads at the University of Beirut. Then she proceeded for PhD studies at molecular and cellular biology at the University of Strasbourg. And later on, she moved onto a postdoctoral position at the University of Münster, where in 2015, she started her own lab. Since then, she has presented us with what I find fascinating work using different sensors and techniques that allow her to detect changes in metabolic activity at the cellular resolution. And this with the goal to learn more about metabolic homeostasis in the nervous system. And today, this is what she will tell us about. So we'll hear about glia and neuron metabolic interactions in Drosophila, which is actually my favorite organism. So yeah, the stage is yours. Looking forward to hearing it.
So let me see. Can you see it when I change the slide now?
Yes,
okay.
all
yes.
going smoothly.
Okay, great. So thanks for the invitation. I'm very happy to be here. I'm actually going to talk mostly about unpublished data we gained in the last years. And I'm going to talk quite a bit about what we know about carbohydrate transport at the blood-brain barrier of Drosophila, and then also, of course, about glia neuron metabolic coupling in the Drosophila nervous system. So as a short introduction, as you might all know, neurons are very energy demanding cells. To function, they need a lot of energy, but they also need a very constant extracellular milieu. So they are very demanding when it comes to their surroundings. But luckily, they are not alone in the nervous system, but we have glial cells here. I just depicted oligodendrocytes and astrocytes in the nervous system as well. And then at least in the mammals, we have blood vessels to provide the brain with nutrients. Now, since the neurons need a very defined extracellular milieu, we have the blood-brain barrier that is in mammals established by the endothelial cells that build the blood vessels. And this actually prevents paracellular diffusion of nutrients into the nervous system. So we have nutrients like carbohydrates, for example, in circulation, but they cannot freely diffuse into the nervous system. Thus, we need to have a variety of transport mechanisms established at the blood-brain barrier to fulfill all the metabolic needs the neurons have. Now, we are working in Drosophila on the questions how nutrients enter the nervous system and then how they are metabolized in the nervous system. You all might have seen such a fly before. Not so sure if everyone who listens has seen a fly brain before. And actually, can you see my mouse? No? Can you? Can you see the mouse, Peter? No? Okay, I'm not sure if you can see my mouse. Probably not. So this is a brain of a fly. So the upper part is the brain and the lower part is the ventral nerve cord. That is the equivalent of the spinal cord in mammals. And when you work with Drosophila, you don't just get the adult fly, but you also get the larva as a basically additional model system. In the larva, you have the central brain and the ventral nerve cord fused together. So here, the central brain is at the left and the ventral nerve cord is at the right side. Can you actually see the mouse? No, I think we lost Rita now, so I don't know. Maybe you can see it now. We'll see. So the Drosophila nervous system is composed of several cell types, as also the mammalian nervous system is. And we have several glial cell types, but we of course also have neurons. And the neuronal cell bodies are found in the cortex region, and they project their axons and dendrites into the neuropile region that in Drosophila is completely cell body free. And then we have the astrocyte-like glial cells. We have the enshithing glial cells and the cortex glial cells that surround either the neuropile or are found in the cortex of the Drosophila nervous system. And then we have two glial cell layers that build the blood-brain barrier in Drosophila, the perineural glial cells and the subperineural glial cells, of which the latter build pleated septate junctions that prevent paracellular diffusion. Now, since the Drosophila nervous system is not vascularized, but Drosophila has an open circulatory system, this means that all organs, including the brain, basically float in the hemolymph, that is the blood-like fluid in insects. We don't have vascularization of the nervous system, but we have a blood-brain barrier surrounding the whole nervous system, since the nervous system needs to be protected from circulation also here in Drosophila. Now we would have all the nutrients out here and they need to get into the nervous system somehow. Like in mammals, the blood-brain barrier is tight during late developmental stages. And if you have a wild-typic embryo in this case here, and you inject a fluorescent dye into circulation, this usually doesn't reach the nervous system since the blood-brain barrier is tight. But if you have blood-brain barrier defective animals, the dye penetrates into the nervous system, as it would be the case also in blood-brain barrier defective mouse embryos, for example. So this is a diffusion barrier as it is in mammals. And now the question is, how do nutrients get into the nervous system? So that's going to be the first part now, where I'm going to talk about carbohydrate transport. And in Drosophila, in circulation, we have several carbohydrates. We have Trehalose, which is the main circulating sugar in Drosophila. Trehalose is a glucose dimer. Then we also have glucose, and in less high concentrations, we also have fructose in the hemolymph. So we have 3 carbohydrates basically that could potentially be transported into the nervous system. Now, a couple of years ago, when we set out to understand how carbohydrates are transported over the blood-brain barrier in Drosophila, we started out first to ask if glucose is transported in all cell types in the nervous system. So if at all glucose is transported over the blood-brain barrier. So to this end, we used a genetically encoded FRET glucose sensor that works as follows. So you have a glucose binding protein fused to a CFP and a YFP. And if glucose is not bound, the CFP and the YFP are quite far apart. But if then glucose is bound, the conformation changes, and the CFP and the YFP are quite close together. And then if you excite the CFP, you will get an energy transfer from the CFP to the YFP. So when you have glucose, you will get a YFP fluorescence when you excite the CFP. If you don't have glucose, you will not get a YFP fluorescence when you excite the CFP. Now you can measure the CFP, YFP emissions, and then and then calculate CFP emission by YFP emission, and this will give you a proxy of, of how much glucose you have in the cell. So we cloned the sensor and put it in the fly. And here on the right side, you can see how it looks in a live ex vivo larval nervous system, here expressed in astrocyte-like glial cells. And when you then have the ex vivo brains in a flow-through chamber, and you go from a no glucose buffer to a glucose containing buffer, you can see that in all the inner neural cell types, like the astrocytes and sheathing glia, cortex glia, and also the neurons, you get a rise in intracellular glucose concentrations when you put a lot of glucose in the buffer, which is then equal to the circulation in vivo. And what you can't see here, but what we also tried, is that also the blood-brain barrier forming glial cells, the subperineural glia and the perineural glial cells, they also take up glucose from the buffer, which means that all neural cell types can take up glucose. What we don't know if they really also can take up Trehalose, because unfortunately there's no working Trehalose sensor so far that has been used in Drosophila. We might have one at one point, we'll see. But for now, we don't have a Trehalose sensor, unfortunately. So this tells us that all the cell types in the nervous system can take up glucose. The question is, how can they do this? So we started out very obviously, because there are 2 Trehalose transporters encoded by the Drosophila genome, Trehalose transporter 1.1 and Trehalose transporter 1.2. A couple of years ago, they were just called Trehalose transporter because they had high homology to some plant Trehalose transporters, actually. So people thought they might be Trehalose transporters, but now we know that at least TRED11 really does transport Trehalose. And when we started analyzing those genes, what we first did was to make a deficiency that takes out both genes. They are conveniently placed in tandem in the Drosophila genome, so this was easy to get rid of both of them. And a deletion removing both TRED11 and TRED12 is lethal. So this was already good because this told us that we need at least one of those transporters. And then we also made a TRED1 single mutant, and this is also lethal, while TRED1 single mutants are viable. When we knock down TRED1 in just the glial cells using RNA interference, what we see is that the animals can't move properly. And the first thing we did was to use the so-called island assay. And you have an island. And what you do is you just flip the flies onto the island, and the wild type animals, they just fly away immediately because they hate being thrown onto something. So if they can fly, they fly away. TRED1 knockdowns, as you can see here, can't fly. So they stay on the island, and then at one point they jump from the island if you scare them enough. So this was the first hint that indeed we need TRED1 in the glial cells. But of course, RNA interference is always a little problematic because you never know if you have off-target effects, you never know if you have a very good knockdown or if you have a hypomorphic phenotype and things like that. So what we did to really prove that we need TRED1 in the glial cells was rescue experiments. And the first thing we did with rescue experiments was to see if the lethality of this deficiency is due to the loss of TRED1 or the loss of TRED1 or both. Now, for everyone who is not very familiar with Drosophila, just an info slide basically. What we can do in Drosophila is to use a driver to drive GAL4 expression. And this GAL4 then can bind to another transgenic element, to the US sequence, and induce the expression of some transgene to be put under the control of US. In this case, we were using a ubiquitous or tissue-specific GAL4 driver that then drove the expression of TRED1 cDNA or TRED1 cDNA. And when we did this, what we found was when we rescue animals homozygous for this deficiency with a ubiquitous re-expression of TRED1, they were happy, but a ubiquitous re-expression of TRED1 didn't rescue the lethality of the deficiency, telling us that TRED1 is the essential transporter and TRED1 is not essential. Now, to figure out if we need TRED1 in glial cells specifically, we added another player to the system that is a GAL80. GAL80 is an inhibitor of GAL4. So what we did was to express GAL80 in a specific cell type in the background of a ubiquitous GAL4. And then when you have GAL80 in a specific cell type, GAL80 binds GAL4. And in this specific cell type, TRED1 is then not expressed any longer. So we can have animals that have a rescue everywhere but in one specific cell type. So this is what we did. And when we did this and had ubiquitous re-expression of TRED1 in the background of the deficiency with glial GAL80, so we have TRED1 everywhere but the glial cells, the animals are dead. If we have TRED1 everywhere but in the neurons, the animals don't care. So they don't need TRED1 in the neurons, but they need TRED1 in the glial cells. So this told us that TRED1 is an essential transporter in glial cells, but of course we wanted to know in which glial subtype we really need TRED1. So we made an antibody against TRED1. And when you stain, in this case, larval brains, what you can find is that TRED1 here in magenta localizes to the perineural glial cells. This is the outermost glial cell layer in the nervous system that is part of the blood-brain barrier. And this is the only glial cell type in the nervous system that expresses TRED1. Yeah. And of course, then we also wanted to know if TRED1, which carbohydrates TRED1 can transport. So we expressed it in xenopose oocytes. That's frog oocytes, and they are commonly used to analyze transport capacity of transporter proteins because endogenously they don't express any transporters on the cell membrane, but you can heterologously express transporters from different species, and they can also then be localized at the membrane actually. So you will not have any endogenous transporters at the membrane, but if you heterologous express transporters, they will go to the membrane. And then when we did this for TRED11, we see that xenopus oocytes expressing TRED11 can take up glucose as well as trehalose efficiently, but fructose not very efficiently. This indicates that TRED11 is actually not just a trehalose transporter, but can also transport glucose into xenopus oocytes at least. Yeah, so since TRED11 is just expressed in the perineural glial cells, we were wondering if there are other transporters in the other glial cell types, since, as I showed you in the beginning, they all can take up carbohydrates. So we did a small biased RNAi screen where we knocked down quite some genes that were supposed from the amino acid sequence to be probably carbohydrate transporters. And we ended up with a couple of genes that, when knocked down in glial cells, were lethal. And then we went on analyzing in more detail, actually two of them, CG4797 and CG4726. That is also called MFS3. That is Major Facilitator Superfamily Transporter 3. That doesn't tell you much at the beginning, but there was also not much known at this time about those two transporters. After we knocked them down panglially and found that knockdown in all glial cells is lethal, we went for glial subtype drivers to express the dsRNA. What we found that knockdown in astrocyte-like glial cells, cortex glial cells, and in sheathing glial cells had no effects. So the animals were viable. For MFS3, subperineural glial knockdown was also viable, but perineural glial knockdown was semi-lethal. For CG4797, both subperineural glial knockdown and perineural glial knockdown were lethal, indicating that both transporters are expressed in the blood-brain barrier, the MFS3 just in the perineural glial cells where TRED11 is also expressed, and CG4797 in both the perineural and subperineural glial cells. And we also wanted to see if this is true by checking for their expression. And for MFS3, there is actually it's a YFP protein trap. Sorry for this mistake. There's a YFP protein trap available that we stained. And as you can see here in L3 larval brains, you get expression in the surface glial cells and to some extent in the neurons. And when you zoom in and you co-stain with a perineural marker, you see that the YFP and the perineural glial marker co-localize nicely. So this really seems to be expressed in the perineural glial cells. For CG4797, unfortunately, there was no protein trap available. So what we did was to clone the complete locus of CG4797 with the upstream and downstream parts to hopefully include all promoter elements that are essential for CG4797 expression, and then fuse the 3'HA tag to the C-terminus of the transporter, and then put this mini gene into the fly genome on another chromosome than the endogenous transporter. And when we did this, we found that the HA tag is actually not very easy to stain. So I just have an image of an adult brain here, because unfortunately all our HA antibodies don't work very well in immunostainings. But when you co-stain a surface glial marker that stains both the perineural and subperineural glial cells, you find basically complete colocalization of the two. So it seems that the expression, the protein expression, recapitulates the dsRNA phenotypes. So then, of course, we were wondering if those two transporters are really able to transport carbohydrates. So we also put those into xenopus oocytes, and when we stained those oocytes, also for these, we used HA-tagged versions in this case as well. When we stained the oocytes, we saw that also those go to the membrane nicely. And for MFS3, we found, as for TRED11, that it transports trehalose and glucose, but not fructose. You might wonder why they always transport trehalose and glucose. But if you think about the specificity of a transporter, it's not so surprising since trehalose is a glucose dimer. So it's not so different from glucose. It's bigger, but the subunit is the same. So it's probably not so surprising that they can transport both. But MFS3 also is not transporting fructose, which is a different monomer also. And the same actually is true for CG4797, indicating that we indeed have 3 carbohydrate transporters expressed in the blood-brain barrier that can transport both glucose and trehalose. And now since CG4797 didn't have a name yet, but it's a carbohydrate transporter, we called it pippin, after Frodo's friend, who is always concerned about where to find the next meal. So this is now pippin. And we went on analyzing those transporters. And to do so, we generated mutants. And we did this by CRISPR-mediated homologous recombination, where we had 2 sgRNA constructs, that targeted an upstream and a downstream region of each gene. And we injected those together with a homology construct where we had a region upstream of the coding sequence of either pippin or MFS3 and a region downstream of the coding sequence. And between them, we put a mini white. And now if you induce double strand breaks in here and here, you get homologous recombination if you're lucky. And you replace the complete coding sequence of the gene by a mini white. And this worked out fine in both cases. And the good thing about this is that you really remove the complete coding sequence. So you can be quite sure that what you have is a null mutant. And we did this, and what we found was both null mutants are viable. This was a little bit of a shock for my student back then, because obviously she thought since the RNAi is lethal, the animals would be lethal, but they were not. Nevertheless, we decided to have a look at their phenotype. We also made a double mutant, and this is also viable, so they don't compensate for each other. But when we had a look at the survival curves, here in black is the control, and these 2 are the single mutants, and that's the double mutant. What we found is that they are short-lived compared to control animals. Interestingly, the double mutant is longer-lived than the single mutants, which was something that was very puzzling at this time. But what should you do? We went on analyzing the phenotypes and found that they also have behavioral phenotypes. And what we did was to use a Drosophila activity monitor. And here you put a single fly in a glass tube. And you do this several times, so you have many flies. And then you monitor the movement of the animals using an infrared beam in the middle of the tube. And every time the animal crosses the beam, the computer registers this, and then you get a movement profile of the animal. Usually the setup is used to study circadian rhythm in flies, so to see if they are active at dawn and at dusk and not so active at noon and at night. In this case, we didn't care too much about their rhythmicity, but we wanted to see if they move as much as the control animals. And what we found was that when they are 2 weeks old, so not very old for flies, the single mutants as well as the double mutants move significantly less than the control animals. When they are older than 5 weeks of age, the single mutants still move less than the controls, but the double mutants now move significantly less than the single mutants. So they seem to have a progressive loss of activity, while the single mutants do about the same as at the age of 2 weeks. So those animals have some kind of problem. So we wanted to see if the double mutants, they could move in theory as much as the single mutants, but don't, probably to save energy. So we used another assay that is a negative geotaxis assay, where you put the animals in a vial and then you tap them on the table, which induces an escape response of the fly. So this is a different thing than voluntary movement. And at 2 weeks of age, all these animals can move as well as the wild type, if they have to, if they want to escape. At 5 weeks of age, however, the single mutants as well as the double mutants move significantly less than the wild type. So in this case, we think that they really cannot move as fast as the wild type, because if you don't try to escape, you usually do it because you can't and not because you don't want to. So we think that in the activity monitor, why all these mutants do not move as much as the wild type at this early time point is because they probably try to save energy. But if you force them to move, they can still move. Yeah. So we were wondering if, since the RNAi phenotype is lethal, if we have compensatory mechanisms in place that somehow rescue the mutants. And what you basically have two possibilities to rescue the loss of a transporter. That is either increasing the concentration gradient between circulation and the cell cytoplasm, because all those carbohydrate transporters are facilitative transporters. That means they facilitate carbohydrate uptake along a concentration gradient, and the steeper the gradient, the more carbohydrates are taken up. So you could change the steepness of the gradient, or you can have more transporter. That would also induce more uptake of carbohydrate into the cell. So we tested actually both hypotheses. And first we checked circulating glucose levels in those animals. And indeed, we have higher free glucose levels in the hemolymph of the single mutants as well as the double mutants. So this is always all significant to the control, but within those, the differences are not significant. So they seem to have about the same circulating glucose levels. So this could be one aspect of why the mutants are not lethal, because they have higher circulating glucose levels. In addition, in the background of the mutants, we checked the expression of TRED11, since we have a TRED11 antibody. This is the other carbohydrate transporter I told you about. And compared to control animals, here you see the tip of a ventral nerve cord of a L3 larva. You can see that you have more TRED11 in the pippin, MFS3, and MFS3 mutants, as well as in the double mutants. And when we quantify this, this is significant, and you have about 2.5 to 3 times as much TRED11 fluorescence than in control animals. So you have a significant upregulation of TRED11 in those mutants, indicating that the TRED11 upregulation could rescue the loss of either MFS3, pippin, or both. Now, of course, we wanted to know why we have a different phenotype in the knockdowns. And when we stain TRED11 in a glia-specific knockdown of pippin or MFS3, we found that we don't get any compensatory upregulation of TRED11, indicating that compensation of a acute knockdown, that is basically RNAi always depletes the mRNA, but the mRNA is produced in the first place. The compensation then is completely different than the compensation in a null mutant. So this is something one should keep in mind when working with RNA interference, because it seems to be differently compensated than null mutants. So we don't have TRED11 upregulation in knockdown animals. Now, of course, we also wanted to see if this really has an effect on the uptake of carbohydrates into the perineural or subperineural glial cells. So we first analyzed glucose uptake in perineural glial MFS3 or pippin knockdowns. So the perineural glial cells, as I said before, are the outermost layer of the blood-brain barrier. And when we had a look at pippin knockdown in the perineural glial cells and put the glucose sensor also in the perineural glial cells, what you can see is that glucose uptake into the glial cells when you have a pippin knockdown is less efficient than in control animals. The same is true when you have an MFS3 knockdown compared to control animals. And you can quantify this. The maximum glucose concentration you reach in the cells is lower in the pippin knockdown as well as in the MFS3 knockdown. So it seems that indeed MFS3 and pippin are transporting glucose into the perineural glial cells, at least in ex vivo brains, as we have here. Now for the subperineural glial cells, pippin, as I said, is expressed in both the perineural and subperineural glial cells. So when we knocked it down in subperineural glial cells and expressed the glucose sensor in parallel, we see that the uptake efficiency into the subperineural glial cells is reduced. In contrast, MFS3 knockdown doesn't do much, which is not surprising since MFS3 doesn't seem to be expressed in the subperineural glial cells. And also here you can quantify the glucose uptake efficiency when you have a look at the slope. Here is less in pippin knockdowns, and also the maximum plateau is lower in pippin knockdowns. MFS3 knockdowns in subperineural glial cells are indistinguishable from controls, which makes sense, as I said before, since it's not expressed in this cell type. Now, of course, the question was, do we see a difference between the mutants and the knockdowns? So next, we had a look at the mutants and also analyzed glucose uptake efficiency into the perineural glial cells first. And here in the pippin mutants, we don't see a difference to control animals. The same is true in MFS3 mutants. Glucose uptake is indistinguishable from the controls, indicating that indeed the TRED1 upregulation and the higher circulating glucose levels rescue the carbohydrate uptake into the perineural glial cells. In the double mutant, however, it seems that at least the uptake rate is not rescued, since the slope is significantly smaller in those cells than compared to controls, but eventually they still reach the maximum glucose concentration in the cytosol. So there might still be a small defect in the double mutants. This might also be why they move even less and try to save energy, which then in turn might make them live longer, which is kind of interesting by itself. But the single mutants are completely rescued by the TRED1 upregulation and the higher glucose circulating levels. In the subperineural glial cells where just pippin is expressed, what you see in the mutants is pippin mutants have a lower carbohydrate uptake in the subperineural glial cells. And here what I didn't show you is TRED1 is usually not expressed in the subperineural glial cells and is also not in pippin mutants. So the compensatory upregulation is just found in the perineural glial cells. So loss of pippin does not seem to be rescued in the subperineural glial cells, which is also reflected by the problem in carbohydrate uptake. And in MFS3 mutants, you don't have any defects in subperineural glial cells, which is not surprising because this transporter is not expressed there. In the double mutants, here we see exactly the same phenotype as we have in the pippin single mutants, but this makes sense. If you just combine those two phenotypes, you end up with the pippin phenotype. And also the slope, again, is significantly less steep than in the controls, and the plateau that is reached is less high than in the controls. So pippin is essential to take up glucose into the subperineural glial cells, and both pippin and MFS3 are needed to take up glucose into the perineural glial cells, but the mutants are compensated, which is not the case for the RNAi knockdowns. So since TRED1 is upregulated upon loss of pippin and MFS3, we decided to have a closer look at TRED1 regulation. So we were wondering if just loss of another transporter can induce TRED1 upregulation, or if also some other problems, let's say, can induce TRED1 upregulation. So what we did was to starve the animals, because starvation induces a drop of circulating carbohydrate levels. So what we see in the ventral nerve cord and also in the central brain of L3 larvae is when we starve them, we get a higher expression of TRED11. And this expression, this induction of TRED11 seems to be induced by the circulating carbohydrates, because when you give them sugar but nothing else, no proteins, no anything, you rescue this phenotype, which you can also see here when you have the relative TRED11 fluorescence in starved versus fed animals. It's about 3. And when you have sugar fed versus fed animals, it's around 1, so you don't get any TRED11 induction. Then we also were wondering if TRED11 is transcriptionally or translationally upregulated. And what we did was to clone the TRED11 promoter and fuse it to a Stinger GFP. And when we starve animals that harbor this construct, so TRED11 promoter fused to GFP, we see that the GFP expression goes up when we starve the animals, indicating that TRED11 is transcriptionally regulated upon starvation. So, of course, the question was, does this have an effect really on carbohydrate transport into the blood-brain barrier forming glial cells? So we used our glucose sensor again. And what we saw was that indeed in starved animals, we have much more efficient glucose uptake than in fed animals into the blood-brain barrier forming glial cells, as you can see here. So indeed, this TRED11 upregulation promotes glucose uptake into the cells. So we wanted to know, how is TRED11 upregulated? Now, if you think about circulating carbohydrate levels, the first thing that comes to your mind is insulin signaling. So we knocked down the insulin receptor in the blood-brain barrier of Drosophila. No, sorry, it's not a knockdown, it's dominant negative insulin receptor expression in the blood-brain barrier forming glial cells. So you basically abolish insulin signaling in those cells. But when you then have a look at those animals, the fed levels of TRED11 are the same, and they all can upregulate TRED11 upon starvation. And when you quantify this and you have a starved versus fed ratio, it's the same in the control animals that express GFP as well as in the insulin receptor and dominant negative animals. So insulin doesn't seem to be regulating TRED11 expression. Now, the counterpart of insulin in insects is adipokinetic hormone, AKH. So we used AKH mutants and analyzed expression of TRED11 in the fed and starved state. But also here, we have an upregulation still in starved animals. And also here, we don't have any differences between the control and the mutants. So also AKH signaling does not seem to control TRED11 expression. Now, another pathway, signaling pathway that is regulating circulating carbohydrates, but not just that, but also nutrient availability in circulation in general, is the TGF-beta signaling. Now, TGF-beta signaling is working like this. You have the activin and the BMP branch with several ligands that can bind to type 1, type 2 receptor complexes, and then activate a phosphorylation cascade that then induces transcription of target genes via SMART mid-air or MAT mid-air complexes. Now we have in mammals as well as in Drosophila those 2 branches, and they share the type 2 receptors, in this case in Drosophila PAND or wishful thinking. So we first knock down PAND or wishful thinking in the blood-brain barrier forming glial cells to see if then we can still get an induction of TRED11 upon starvation. And what you can see here is you have here 2 different RNAi lines against PAND and 1 against wishful thinking. And in the PAND knockdown, we don't see an upregulation anymore of TRED11, indicating that indeed, TGF-beta signaling via PANT, not via wishful thinking, since here you still have the upregulation, is needed to upregulate TRED11. Now, you also have type 2 receptors that are for the activin branch, baboon, for the BMP branch, either saxophone or thick veins. So we also wanted to know if we need those to upregulate TRED11. And when you have a look at baboon animals, fed or starved, you still have an upregulation when you lack baboon in the blood-brain barrier. For saxophone knockdowns, you also still get an upregulation, but you don't get an upregulation of TRED11 when you lack thick veins. So it seems that TRED11 is regulated via the BMP branch of TGF-beta signaling, via the receptors thick veins and PANT. So of course, we also quantified this. You don't need to believe just in the stainings. So in the case of PANT knockdown, we don't get this induction of TRED11 anymore if you have starved versus fed. And also in the case of thick veins knockdown, we don't get the induction anymore. Now, it seems to be, as I said before, the BMP branch of TGF-beta signaling. Now, as you can see up here, you have several possible ligands for this receptor combination that are screwed, glass bottom boat, and decapentaplegic. So we wanted to know if one of those ligands actually can induce TRED11 upregulation. Now, if in the fed state, you would assume that you don't have these ligands because you don't have an upregulation of TRED11. However, in the starved state, you seem to have those ligands and then get TRED11 induction. So what you would expect is that if you overexpress those ligands in the fed state, you should get an artificial induction in TRED11. Oh, I lost a slide. In TRED11 expression. And the slide that I somehow lost shows you that indeed, if we express glass bottom boat in fed animals, you get an induction of TRED11. So it seems that glass bottom boat via the receptors PANT and thick veins can induce TRED11 regulation. Yeah. So to sum up the carbohydrate transport part, we have 3 carbohydrate transporters that we know by now that are expressed in the blood-brain barrier forming glial cells and Drosophila. These are TRED11, MFS3, and pippin. And TRED11 and MFS3 are expressed in the perineural glial cells only, and pippin is expressed in both the perineural and subperineural glial cells. Then I showed you that all those transporters can transport trehalose and glucose in xenopose oocytes. With the glucose sensor experiments, I showed you that all of them can transport glucose in ex vivo brains. I further showed you that the transporter expression actually is very dynamic, and loss of transporters can be rescued by compensatory upregulation of another transporter, and that in case of TRED11 upregulation upon starvation, this upregulation is regulated by TGF-beta signaling. One important thing as a take-home message is that the compensatory mechanisms induced in a dsRNA-mediated knockdown seem to be very different than those induced in null mutants, since, as I told you here, we see compensatory upregulation of TRED11 in the null mutants, but not in the dsRNA-mediated knockdowns, which leads to much more severe phenotypes using RNAi than when you have mutant animals. Now, as promised, I'm also going to talk about the cellular metabolism in the nervous system. And a couple of years ago, what we did was to have a look at glycolysis in glial cells and neurons. When you want to use carbohydrates for gaining energy, you need to break them down. And this is done via glycolysis. As you can see here, you might know it from the textbooks, from glucose to pyruvate, but you can also fuel it with trehalose. You just need to break down trehalose into 2 molecules of glucose, and then it's the same cascade. Now, what we did was to knock down all glycolytic enzymes, or several glycolytic enzymes, first ubiquitously. And in case of these 6 genes, the ubiquitous knockdown was lethal, indicating that the RNAi was efficient. Then we did tissue-specific knockdowns in glial cells, and we found that depending on the strength of the RNAi, we either get lethality or severe locomotion defects and short-lived animals. And when we then knock down those glycolytic genes in neurons, we found that nothing happened. The animals hatched and they were happy. So we used 2 different neuronal drivers and we got the same phenotype. So we concluded that it seems that glycolysis is essential in glial cells, but not in neurons. Now, in this case, we had driven the dsRNA constructs through development, and we wanted to know what happens in a fully developed steady state nervous system. So we just expressed the dsRNA constructs in the adult fly. Now, when we did this here, I just depicted Trehalase knockdown and pyruvate kinase knockdown. Otherwise, this would have been twofold. And the control was GFP knockdown. And what you can see when you have a glial knockdown, that's the solid lines, the lifespan is severely reduced compared to controls, while when you have a neuronal knockdown, the lifespan is not affected. So also in the adult nervous system only, glycolysis seems to be essential in glia, but not in neuronal cells. Now, of course, we wanted to know what happens in those nervous systems. Why are these animals dying? And so we did semi-thin sections of the adult heads. Here we have the glial knockdown of Trehalase in this case, and the neuronal knockdown of Trehalase. And what you can see here in dark is the cortex regions, and those lighter regions are the neuropile regions of the nervous system. And in case of a glial knockdown, you see that you develop these huge holes in the cortical regions that you never see in control animals nor in neuronal knockdown animals. And we wanted to know why we have those holes. So what we did was to do activated caspase-3 stainings. Activated caspase-3 stains, these antibody stains, apoptotic cells. And what we found was, in the case of glial knockdown of glycolytic enzymes, we get elevated levels of apoptotic cells. And when you have a look at the type of cells that is apoptotic, you find that it is all neuronal cells. So when you lose glial glycolysis, you lose neurons, indicating that indeed glial glycolysis is essential for neuronal survival. Of course, this means that the glial cells under normal conditions need to provide the neurons with some kind of metabolite, since the glial glycolysis is so essential. And to find out what kind of metabolite this is, what we did was to express GFP in either the glial cells or the neurons, and then dissociate the brains and FAC sort them. And then we ended up with either just primary glial cells or primary neurons. And those we cultured and fed them with a heavy labeled Trehalose for a certain time. And then we analyzed the supernatant via HPLC and mass spec. And what we found is in the supernatant of the glial cells, you find heavy alanine and heavy lactate and heavy glucose. Heavy Trehalose we put in. In the supernatant of the neurons, you just find the heavy Trehalose, indicating that the glial cells secrete alanine and lactate. And in an independent set of experiments, we showed that the neurons can metabolize both alanine and lactate to CO2. So they can take up alanine and lactate and can use them to gain energy. Now, this system is very reminiscent of the mammalian astrocyte neuron lactate shuttle hypothesis, indicating that indeed here we also have a metabolic division of labor between glial cells that are glycolytic and neuronal cells that are oxidative mainly, and that this division of labor is essential actually. So this means also that it's not an adaptation to the huge size of mammalian nervous systems as long thought before, but it's more a basic principle of nervous system function, that you have a metabolic coupling between glial cells and neurons. And since here in this primary cell culture assay, the glial cells were producing lactate, we wanted to see if they also do this in ex vivo brains. So we used a FRET lactate sensor that is working basically the same way as the glucose sensor, and expressed it in the glial cells. In this case, what I'm going to show you now is the sheathing glial cells. And in parallel, we expressed a temperature-sensitive ion channel in the neurons that allows you to activate all neurons at once when you higher the temperature from ambient temperature to 29 degrees. Or 37 is actually better. I think we used 37 degrees. So when you do this, what you can see, no, it was 29, sorry. When you do this in control animals that don't have the TRPA1, you don't see anything happening. But in those animals where you have neuronal overexpression because the neurons express TRPA1, you see that the lactate levels in the glia rise. So the glial cells produce more lactate. So also in Drosophila, as it has been postulated in mammals, this lactate provision seems to be regulated by neuronal activity. Yeah, so this is conserved. And as I said, reminiscent of the ANLS. So this is a conserved principle of CNS function. Now, as you might have noticed when I showed you this survival graph, is that when we knock down glycolytic enzymes in the adult glial cells, those animals live up to 4 weeks, actually, which is half the lifespan of a wild-type fly. So they don't die immediately. So we were wondering if they can compensate to some extent for the loss of glycolysis in the glial cells. So what we did was to have a look at another pathway that can, of which you can produce energy, and this is the oxidation of lipids via beta oxidation. So you have this mitochondrial beta oxidation that is also quite complex. And what we did was to knock down either CPT2 or MTP alpha. CPT2 is needed to transport acyl-CoAs into the mitochondria, and MTP alpha is needed further downstream in beta oxidation. Most data I'm going to show you is going to be the CPT2 knockdown, but the two knockdown phenotypes are indistinguishable. So I'm not going to show you data for both, but they have both the same phenotypes. So what we did was to compare animals with a glycolysis-only knockdown, in this case pyruvate kinase RNAi, or a beta oxidation-only knockdown, CPD2 RNAi. And here we also always added mCherry RNAi since we wanted to compare the phenotypes to the double knockdown. And what happens when you have two UAS dsRNA constructs is that the effect of the GAL4 that you express in the glial cells is diluted between the two UAS constructs. So that's why you need two constructs in each fly to be able to compare the phenotypes. So here you see the glycolysis only knockdown. They now live a little longer than before, where we didn't have the mCherry RNAi, since we have a hypomorphic phenotype. As a control, we had GFP cherry knockdowns. And when you have a look at the pyruvate kinase, CPT2, so glycolysis, beta oxidation double knockdowns, what happens is that this severely reduces life expectancy of the animals, even though you add up two hypomorphic phenotypes here. In contrast, the beta oxidation only knockdown doesn't have any phenotypic consequences in the lifespan. When we do the same, but use a neuronal driver, so do the knockdowns in neurons, nothing happens. They all live the same amount of time. So this indicated that indeed, if you have a loss of glycolysis, the glial cells can compensate for this by using lipids to gain energy. Now we wanted to see if then we also get neurodegenerative phenotypes. So we had a look at them, at the brains of those animals at 17 days. And when we go back once, this 17 days is here just before the double knockdowns die. What you see is in the control, GFP cherry knockdowns, everything's fine. The cortex region looks fine. The pyruvate kinase cherry knockdown at this stage also looks fine, but this is long before they die. So here we also don't have any effects, but in the double knockdowns, we see that we develop those holes in the cortical regions. The beta oxidation only knockdowns doesn't have any problems. If we then have a look at a later time point, that is then shortly before the pyruvate kinase single knockdowns die, we see that Then we also find these holes in the glycolysis only knockdowns, as one would expect, since we showed this before, just without the cherry RNA. The double knockdowns are dead by then, and the beta oxidation only knockdowns still don't develop any phenotypes. So indeed, a glycolysis beta oxidation double knockdown seems to accelerate the rate of neurodegeneration compared to glycolysis single knockdown. Now we wanted to know if also the animals move less because they have a neurodegenerative phenotype. So we again put them in the Drosophila activity monitor. What you can see is, and here we have both double knockdowns, that at 1 week of age already, the beta oxidation glycolysis double knockdowns move significantly less than the control animals and also then the glycolysis only knockdowns. Now, if you have a look at them later on, where the double knockdowns are dead, the glycolysis only knockdowns also move less. So they have progressive neurodegeneration here. Yeah, now, I don't know how much you know about mitochondria, but it has been stated before that in a more fused state, mitochondria favor beta oxidation, and in a more fragmented state, they favor glycolysis and respiratory chain. So we wanted to know if in our glycolysis knockdown glial cells, we see the mitochondria in a more fused state that would really favor beta oxidation. So what we did was to express a mito-GFP in the background of either lactate dsRNA as a control or of pyruvate kinase dsRNA. And what you can already appreciate from these 3D images is that the pyruvate kinase knockdown leads to more fused mitochondria. And we can also quantify this by putting the total mitochondrial volume in relation to the number of volume units you have. And what you can see that in the pyruvate kinase knockdowns, you have significantly more fused mitochondria than in the control animals, indicating that indeed also the mitochondrial shape indicates that we have beta oxidation going on in the mitochondria. Now, we wanted to know what do those glial cells produce. Now, what you can produce from beta oxidation is ketone bodies. And it has been shown that the nervous system can use ketone bodies, at least in mammals. So we did another set of survival experiments where we knocked down ketone body metabolism in addition to glycolysis instead of beta oxidation in addition to glycolysis. And as you can see, also additional loss of ketone body metabolism in the glial cells reduces lifespan severely compared to glycolysis only knockdown, indicating that the glial cells, when they can't use glycolysis to fuel the neurons, indeed produce ketone bodies to fuel neuronal metabolism. And also these animals are significantly less active than the control animals already with 1 week of age, while the ketone body only knockdown doesn't have a phenotype. Thus, and since of course you would expect if you have beta oxidation that the animals use lipids, we did lipidomics and we indeed found changes in lipids in the fat body of the animals. And now here I'm talking about the fat body in a glia-specific knockdown. So I'm not talking about triacylglyceride levels in the brain, but in the head fat body, which is the periphery, which is the storage organ. And what we see is indeed that when we have changes in glial metabolism, we influence the amount of triacylglycerides that is stored in the fat body. So what happens is that we must have some kind of signaling between the brain and the periphery that says you need to change systemic metabolism, but we just have a knockdown in glial cells. So it's not a ubiquitous change in the brain. It's not a neuronal knockdown. It's just a glial knockdown, and we still get systemic effects. We get changes in triacylglyceride in the head fat body, and we get changes in diacylglycerides in the hemolymph. So somehow, in the case of changes in glial metabolism, the nervous system signals to the periphery that there is a problem and to adapt the systemic metabolism. Now, this is data we have now, and we have no idea by now how this works. But there is some kind of signaling. I can tell you that it's not insulin and that it's also not AKH, which are the obvious players again. But we will still need to figure out in the future what's the signaling here. How do the glial cells tell the periphery that there is a problem in metabolism, even though they still manage to keep the neurons happy? And with this, I'll come to the second summary. So the glial and neuronal metabolism are coupled in Drosophila. We have the same coupling as in mammals, and this indicates that this is a basic feature of complex nervous systems. The glial cells produce, under normal circumstances, lactate and alanine that they shuttle to the neurons. If the glial cells cannot use carbohydrates to fuel neuronal oxidative metabolism, they switch to beta oxidation and produce ketone bodies to fuel neuronal metabolism. And changes in glial metabolism induce systemic changes in metabolism in the fly. And with this, I want to thank the people who did this work, my PhD students, master students and technicians in the lab, former PhD students, and all my collaboration partners, and of course, also the funding. And of course, I thank you for your attention. And I still can't see Rita, so I'm not sure what's going to happen now. Yeah. There you are.
Okay, finally. Okay, sorry. I'm so sorry, everyone. We also got a bit delayed because of this troubleshooting. Okay, so thank you so much for the talk. It was really interesting. We learned a lot about these transporters and brain metabolism. So we can move on to the questions since we are running a bit late to go then to the Q&A, where if anyone actually wants to join, drop us an email. I will put the email here, and we can check if we still have room for some more people. Okay. So the question that we have here is from Anthony Bayenkamp-Salazar, and he says, "Thank you for the very nice presentation, Dr. Schirmeier. Would this knockout on pippin or MSF3 also affect any feeding behavior? Did you see longer feeding times or feeding times in spite of the lower motility?"
We actually didn't do any feeding analysis. I mean, the RNA knockdowns, they are pupil lethal, so you would need to go for larval assays here. We didn't do this. In the mutants, in the adult, I actually don't know. We didn't do this. We know that we have higher circulating carbohydrate levels in the mutants. We have more tridon-1 in the mutants, but I don't know if they compensate by feeding or if they compensate by mobilizing carbohydrates from the fat body. This I don't know. We never checked. We should do this, actually. I think we lost her again. Maybe you can also put questions in the chat and then I can repeat them while we wait for Rita. Maybe? So Sophia is asking if for the longevity experiments, it doesn't matter what the dietary composition is and when the food is administered. I'm not sure if you mean the transporter loss or the glycolysis knockdowns. Actually, in case of the transporter mutants, so MFS3 and pippin mutants, we tested carbohydrate-rich food, and in this case, they are also short-lived. So this doesn't change much. We tried very lipid-rich food, but they actually didn't eat it. So what we saw was a starvation effect, which doesn't really help. So for carbohydrate-rich food, it doesn't seem to make a difference. They are still short-lived compared to the control animals. For the beta oxidation knockdowns, actually, they seem to be susceptible to starvation. Follow-up question, are there sex differences? We actually, for the glycolysis beta oxidation knockdowns, we just use females because the pyruvate kinase RNA is on the X, which makes it easier. For the transporters, I think we also just used females. I don't know if males would react differently. Another question is if for our labeling experiments concerning the glycolysis stuff, if we have higher labeled lactate and alanine levels in the neurons. Actually, we didn't do any co-culture experiments. So we just either cultured the primary glial cells and then had a look what we find in the supernatants, so what they secrete, or we fed cultured neurons with labeled alanine or lactate. But we never did co-culture experiments. I mean, I would guess this should happen, but we never did those experiments. Another question is if the cortex and in-sheathing glia are also involved in shuttling lactate and alanine towards neurons. Actually, all the glycolysis and beta oxidation knockdown experiments we did was using a pan-glial driver. We actually tried astrocyte-like glial drivers and cortex glial drivers as well. We didn't see big effects, but this might have two reasons. First, the subtype-specific drivers are less strong than the pan-glial driver, so you might have a weaker knockdown. And second, it might be that if the astrocyte-like glial cells, for example, lose the ability to break down carbohydrates, the other glial cells might compensate. This we can't really say. We just know that the strongest phenotype is with the pan-glial driver. But comparing the different drivers is very difficult because they don't have the same strength. So it might be both. I don't know. So someone is also asking if alanine and lactate could expand the lifespan of CPT2 knockdown flies. We tried. We tried feeding lactate, we didn't inject it into the hemolymph or something. This didn't do much. but we are not sure how well lactate is taken up over the gut epithelium. So we don't know if it really reaches the nervous system. We also tried feeding beta-hydroxybutyrate, which is a ketone body, but also there we don't know how well it's absorbed. But we didn't see any differences when we fed the flies with those compounds. So I guess we are going to stop here. Then I think I'm going to go to the Zoom. I don't know how to end this. Thanks all for listening.